Question number 171. Select the correct statement with respect to SCID. SCID stands for Severe Combined Immunodeficiency. And the very first statement is caused due to absence of gene for adenosine deaminase. And as it is a correct statement, so this is a correct answer to our question, because severe combined immunodeficiency is caused due to absence of gene for adenosine deaminase. Next statement is cannot be treated by bone marrow transplantation at early stage, and this is an incorrect statement because this disease can be treated by bone marrow transplantation at early stage. The next statement is in presence of adenosine deaminase, lymphocyte proliferation is inhibited, and this is an incorrect option because not in presence but in absence of adenosine deaminase, lymphocyte proliferation is inhibited. The last statement is without RBCs, ADA deficient children are wide open to attacks of viruses and bacteria, and this is also an incorrect statement because without T cells or WBCs and not RBCs, ADA deficient children are wide open to attacks of viruses and bacteria. So the correct answer to this question is option number 1. Let's discuss our next question. Question number 172. The main challenge for production of insulin using recombinant DNA techniques was getting insulin assembled in mature form by, and the options are obtaining correct sequence of amino acids in chain A and B. Second option is removal of chain A from proinsulin. Option 3 says formation of disulfide bonds between correct amino acids, and the last option is transformation of E. coli with cDNA of chain A and B. And we know that removal of chain C is required to form mature insulin from proinsulin. Eli Lilly, an American company, prepared 2 DNA sequences corresponding to A and B chains of human insulin and introduced them in plasmid PBR322 and then transformed E. coli to produce humulin. So there is no problem in transformation or in formation of correct sequence of amino acids of chain A and B. So the very first statement, obtaining correct sequence of amino acids A and B, is an incorrect option, and option 2, removal of chain A from proinsulin, is also an incorrect option because it is removal of chain C from proinsulin that will produce mature insulin. And the last option, transformation of E. coli with cDNA of chain A and B, is also an incorrect option. And the correct answer to this question is option number 3, formation of disulfide bonds between correct amino acids, because disulfide bonds are formed between 2 cysteine residues on chain A and B in correct orientation, and it is the main challenge to produce insulin using recombinant DNA techniques. So the correct answer to this question is option number 3. Let's discuss our next question. Question number 173. The term used to refer to the use of bioresources by multinational companies and other organizations without proper authorization from the countries and people concerned without compensatory payment is, and the options are biopatent, bioethics, bioweapon, and biopiracy. And the correct answer to this question is option number 4. Biopiracy is the term used for the use of bioresources by multinational companies and other organizations without proper authorization from the countries and people concerned without compensatory payment. Let's discuss rest of the options. The very first option given is biopatent, and this is an incorrect option because patents which are being granted for biological products derived from them are known as biopatent. The next option is bioethics, and this is an incorrect option. Bioethics may be viewed as a set of standards that may be used to regulate our activities in relation to biological world. And the next option is bioweapon, and this is an incorrect option because bioweapons are biological agents such as bacteria, fungi, viruses, etc. with an intent to kill humans, plants, animals as an act of war. So this is also an incorrect option, and the correct answer to this question is option number 4. Let's proceed to the next question. Question number 174. Choose the odd one with respect to technique used for early detection of diseases. And the options are enzyme-linked immunosorbent assay or ELISA. Second option is recombinant DNA technology. Third option is urine analysis. And the fourth option is polymerase chain reaction. And we know that modern methods of diagnosis serve purpose of early diagnosis, which are recombinant DNA technology or RDT, ELISA, PCR, etc. So these are the incorrect options. Whereas urine analysis is a conventional method of diagnosis and is a correct answer to our question. It is a technique which is not used for early detection of diseases nowadays. Whereas other three options are modern methods for early detection of diseases. So the correct answer to this question is option number 3. Let's proceed to the next question. Question number 175. How many recombinant therapeutic are currently being marketed in India? And the options are 10, 12, 15 or 30. And the correct answer to this question is option number 2, 12. Whereas rest of the options are incorrect. 10 and 15 are incorrect options and 30 is an incorrect option. Because at present, about 30 recombinant therapeutics have been approved globally and only 12 of these presently being marketed in India. So the correct answer to this question is option number 2, 12 and not 30. Let's discuss our next question.